I have to write like summaries of 2 capstone papers, summaries of 2 post-presentations, and then like a summary for the
One thing you learn is the title. That's it. Like 2 capstones. All of them learn one thing. You can like notes, but then and then in the prompt or whatever, she's like, don't write an essay or paragraph if you're doing local credit. Dang. Like writing like Jesus in there, you know what I mean? Like that's crazy. She probably just did it like story. Yeah. So like it's amazing. It's not that all she has like 3 things she has like threshold and then there's like 1 bullet research and then so she has like just credit
I wish.
that's what I'm saying. Like it's like 2 minutes, but it's
But I mean, I've gotten
like I need to stay here anyway, so like let me just like wish that. You're like
Might as well at that time.
and yes, correct, but we don't want to say anything about it or anything, and then she actually for her capstone, so she actually talked about we were like, let's go.
I love stress. I love how so cool because I had friends and like it was always
Yes. Okay. Actually, 4 for this. But yeah. Then I didn't do anything to the posters. Yeah. I love to.
I was like, I saw one guy, I was like, and then I saw him. I don't know, like, I haven't seen my yeah, I was
Because also she said like in the back for 10 minutes.
yeah, and I was like, something that I comment in from my fire, it did like the shower.
Yeah.
Like, and she's
I'm gonna show her like take a picture of it and put it on my card. And on the top. She thinks I'm here. I didn't do this, and I am. Yeah. And I was like, okay, texting her. I was like, because one of her like things that she wanted for the proposals, I was there. Like, And so she's like, oh, it's your Sarasota. And I was like, well, we have each other's location. So I was like, saying to this that like the symposium is today. So even if she missed that, like she's gonna see that I'm here on Sunday. So she's like, so I texted her and I was like, even though it's like it's a little day, I'm like, oh, well, it's sitting here symposium. And she's like, oh man, that's really unfortunate. My boyfriend has a whole day planned out. Like, that's so late for you, but like, whatever. And I'm like, oh, yeah.
Is that like suspicious for like time of
Really? Yeah. He does have a lot of, yeah. Yeah.
That's so amazing. I'm so excited. I'm excited to do that. I learned this because of my other position. And I told my best friend, well, you're gonna have to before me. So
Yeah.
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 I look for it and like her boyfriend is scared like constantly.
Oh.
But girl,
Yeah.
I'm sorry.
Yeah.
Thank you. I like it.
Only 2 hours. It's okay. It's only 2 hours. I ordered biscuits. It's so funny, guys. Because then if you're mad at them, how do you feel about the
Hi guys, welcome back. We're gonna get started on time. Millie, we're gonna hear you. So I have the privilege of introducing Millie Garcia. She is majoring in biology with a concentration in medical sciences. She's from Puerto Rico and has been at SU for 4 years. While at SEU, she has been a member of PHSO, SCNDA, and served as an SEU clinic volunteer. And she loves this one out. She's also my wonderful TA as well. Upon graduation, she plans to attend Lake Erie College of Osteopathic Medicine in Bradenton starting this summer.
Hi everybody, so as Dr. Adams said, my name is Milianis Garcia. My project today will be on altered thyroid hormone sensitivity and neuroendocrine dysregulation in polycystic ovarian syndrome, or PCOS. I'm going to refer to it as PCOS for the duration of this presentation. So PCOS is a hormonal endocrine disorder that is characterized by hyperandrogenism, so that we see that in like high testosterone. And then we also see this in ovulation dysfunction, so that's anovulation, not being able to ovulate and things like that, and also in neuroendocrine disorders. So this affects about 5 to 26% of reproductive age women worldwide. This is a really wide range because we believe that there are so many women that are currently misdiagnosed, not diagnosed, or ongoing treatment for something different. And then to diagnose PCOS, it has to meet the Rotogram criteria. So we need 2 out of the 3 features that you see right there. So the 3 features are anovulation, biochemical hyperandrogenism, so we see that in high testosterone, and also we can see that in just hirsutism, so that's just hair growth in your face and stuff like that, or acne, and then also we see polycystic ovaries. So PCOS is not just in your ovaries, we see this all throughout the system, it affects your entire system. So I'm just going to mention some of the tops. So we can see that in cardiovascular health risk, obesity. We see it in infertility, and also it leads to mental health issues. So some of the current treatments, there's actually no cure for PCOS. So we treat just the symptoms. So we treat it by lifestyle changes, weight loss, hormonal contraceptives, like oral contraceptives, anti-androgen medications such as spironolactone, fertility drugs like letrozole or metformin. So here we have neuroendocrine dysregulation. I know this seems a little scary, but we're just going to explain this really quickly. So here you see on the left, you have somebody that does not have PCOS, has no neuroendocrine dysregulation. On the right, you see somebody who has neuroendocrine dysregulation. So we'll be focusing on the luteal phase. Your luteal phase is the last 7 days of the menstrual cycle. So as you can see in a healthy person, the gonadotropin releasing hormone, so the GnRH, you see that it has its high frequency in the follicular phase, and then it goes to a low frequency. So that right there is what allows a woman to have the LH surge, so the luteinizing hormone surge, and it allows for the FSH, so follicle stimulating hormone release. And all of this allows a woman to ovulate. So that's what you see right there. On the right, you see somebody that has neuroendocrine dysregulation. They have androgen excess, which antagonizes the ability to decrease GnRH pulse frequency. So you'll see that the peaks are all high. So that right there does not allow for the LH frequency of the LH surge to occur, which then results in anovulation. So that right there is what you see in the ovary. That ovary on the right doesn't go through any of the phases. The ovary on the left goes through all of the phases and eventually sheds. So this is just right here to explain it easily. We do GnRH, pulse frequency is often increased. You see elevated LH and also lower FSH, and all of this contributes to increased androgen production by the ovaries and also an ovulation. So we are not quite sure of what the link is between the thyroid and the PCOS. So that's what we're really looking at. But we think that there might be something right there in that step that can lead to that cascade of effects that we were looking at in the prior slide. So now we're linking thyroid hormone signaling to PCOS. So we know that the thyroid hormone regulates metabolism. So we believe that dysregulation may contribute to PCOS metabolic dysfunction. The thyroid is not just a metabolic regulator, it's also a neuroendocrine regulator. And so individuals with PCOS have a higher likelihood of developing thyroid disorders. And then we see that the evidence suggests a link between thyroid dysfunction and PCOS. So we're just hoping that understanding this relationship may explain persistent hormonal imbalance. So why does this matter? We see that PCOS and thyroid disease share a lot of overlapping symptoms. You can see some of them on that picture on the right. But thyroid disorders increase the risk of PCOS. And so we're thinking that this dysfunction may contribute to fatigue, weight gain, poor coordination, and even memory loss, as well as all of those other symptoms that you see right there. And so understanding this link may improve diagnosis, and we just want to improve overall patient outcomes. So the gaps in knowledge for this study is that we don't really know the role of thyroid hormones in regulating neuroendocrine dysfunction, or function, sorry. And so we still have not studied how the thyroid hormone affects GnRH, LH, FSH surge, ratios, and androgen production, and we do not know the impact of levothyroxine. I bolded it because we're going to see this in the intervention soon enough. So, but we don't know the impact of levothyroxine on hormonal balance, and then we also do not have an optimal treatment strategy for PCOS. And the big question is why women can have subclinical hypothyroidism symptoms with even when blood tests are within normal limits. So now we're going into my specific aims. My specific aim 1 is to identify potential altered thyroid hormone sensitivity in individuals with PCOS. So we hypothesize that women with PCOS will exhibit altered thyroid hormone sensitivity compared to healthy controls. And then the null hypothesis is basically that there will be no difference between both groups. So here's my methodology 1, pretty simple. So we see that it's a case control study. We have 50 patients with PCOS and 50 patients without PCOS. We're using females, reproductive age. And so we see that in 18 to 35, we want to exclude anybody that has officially diagnosed thyroid disease, going through any hormonal therapy, such as oral contraceptives, pregnant, breastfeeding, or trying to get pregnant, and then diabetes. So we're going to measure blood tests. So here we're looking at TSH, T3, T4, LH and FSH and testosterone, as well as the LH-FSH ratio. So this is all done through blood tests. And so then we're gonna just analyze both groups and see the correlation between the thyroid versus reproductive hormones. So now we're going into the specific aim 2. Here we're evaluating thyroid hormone signaling as a potential therapeutic target. So we hypothesize that a low dose levothyroxine will improve thyroid hormone signaling, which will then improve neuroendocrine hormone regulation in women with PCOS. And the null hypothesis is just basically that the intervention will have no effect on the neuroendocrine hormone regulation. So for methodology 2, we're doing a randomized, double-blinded, placebo-controlled trial. So we're doing 60 women with PCOS, 30 receiving a placebo, 30 receiving the intervention. So the intervention is levothyroxine. We're doing 25 mcgs daily. So this is the lowest FDA-approved dose that we can give. So we're just trying to keep everything safe. And then the control is placebo. So our participants, again, females 18 to 35, they have to be officially diagnosed with PCOS. The TSH must be between 2.5 and 4.5. And then we're going to, again, exclude anybody with thyroid disease, pregnancy, breastfeeding, diabetes, or hormone therapy, again, like oral contraceptives. We're going to do all of this by measuring blood tests. We're measuring almost all the same things that we measured in the beginning. So we're doing LH, FSH ratio, TSH, T3, T4, and testosterone. But we're also doing a symptom diary on this one, and I'll show you what that looks like in a second. So we're doing the blood test at baseline and week 12, and we're comparing between groups, within groups, and stuff like that. So for my visual learners, this is what a symptom diary looks like. This is for a different study, but basically, this is just what it looks like, and we're going to be monitoring this throughout the study. So the intervention, why did we choose levothyroxine? So levothyroxine is a synthetic T4 that converts into your active T3 form. So this is standard treatment for hypothyroidism, and we know that it improves thyroid signaling, but we're seeing if it improves it within the normal range. And it enhances T3 and T4 availability. It may normalize GnRH signaling. It lowers LH levels and enzyme production. And again, we're just doing a low dose of 25 daily for 12 weeks. So my expected outcomes for my study is we hope that this will improve thyroid hormone levels, reduce a reduction in elevated TSH. If the clinical hypothyroidism is present, we're going to see a reduction in androgen levels, which may improve acne and hair growth. We're going to see enhanced insulin sensitivity, potential improvements in weight management, and also we're hoping that it has potential fertility benefits. So my significance in my future studies, this is a really significant study. I think it explores understudied thyroid PCOS links. And so we're just hoping that it could personalize treatment for women with PCOS. So it's not just the same treatment for everybody. And we're hoping that it reduces long-term metabolic and reproductive risk. And for the future studies, I would love to do clinical trials on levothyroxine and PCOS, larger trials, and stuff like that. And then studies with larger samples, long-term metabolic and reproductive outcomes, and then also doing combined therapies of levothyroxine, also adding insulin drugs or lifestyle changes. And then I just want to acknowledge God for the ability to be here and do this project, Dr. Adams, my advisor, my professors at ICU, my family, my friends, and my boyfriend. So thank you guys.
So as somebody with PCOS, when you go usually to a doctor, they run all the tests, but so like for me, my thyroid levels were normal, yet you still experience those. So how do you think you could involve the doctor's training, or like how do you think we could educate the gynecology field more on comparing those two together?
Yeah, that's a really good question. I think this is what the study is for. So that's what we're doing this study to see if there is a valid correlation there. And then from there we can share our results. And then we can hope that gynecologists and doctors are more aware that even if patients are on limit, we can maybe start treatment and hope that your symptoms get better.
So I know you said that you would exclude people on birth controls, and because some hormonal birth controls cause anovulation, is there a link between people on birth controls or coming off of birth controls that show up with PCOS?
That's a really interesting question. That was outside of my personal scope for this study, but that's a really good question. I would have to check for you. But yeah, thank you. Yeah.
I was just wondering why we didn't necessarily use the mouse model to test this necessarily.
Yeah, so actually I did start with a mouse model, but I realized that a lot of this is symptom based, not so much about blood levels. Is we're trying to see if in that symptom diary we see improving symptoms throughout. So it just would have been, it wouldn't have been the same with a mouse than with an actual human, and also we know that the intervention is safe. So we thought it'd be better.
So I missed the background information, so I don't know if you said this already, but why would you measure the testosterone levels? Is that affecting PCOS symptoms?
Yeah, so in PCOS you see hyperandrogenism. So that's something like high testosterone. And so that is a really important marker to see that causes hirsutism, acne, hair loss, and stuff like that. So that's also in correlation with what we're studying.
And are there any other correlations to maybe like progesterone or estrogen or something like that?
I definitely could see that being a thing. I think I just wanted to start with simple facts, like just to start out the study. But I think that it would be a really good idea to add that into future studies of doing estrogen and progesterone testing. Thank you for your questions. Okay, thank you guys.
All right, guys, so this is Helen Lavender. She is majoring in biology with a concentration in pharmacy. She's from Riverview, Florida, and has been at SU for 4 years. After graduation, she plans to take a year to work and gain more experience in the healthcare field.
So today I will also be talking about PCOS, specifically the role of inflammatory cytokines in ovulation and induction failure in the PCOS mouse model. So PCOS is polycystic ovarian syndrome, and is the most common endocrine and metabolic disorder that affects 8 to 22% of women. And to be diagnosed, you have to have 2 out of 3 of the Rotterdam criteria, which is anovulation, so not ovulating in your cycle, hyperandrogenism, and which is an increase in male hormones, and polycystic ovaries, which is caused by estrogen underproduction and androgen overproduction. And in this image, you can see on the left is a normal ovary, and the right is a polycystic ovary. The etiology of this disease is unknown, so they don't know exactly what causes it. They have done studies to show that there are multiple genetic and epigenetic factors that go into it. And it is also heavily associated with chronic low-grade inflammation, and it's the leading cause of anovulatory infertility, which are the 2 things that I will be focusing on. So as far as symptoms and treatment go, there are a very wide array of symptoms that vary from patient to patient. Some of the most common ones are irregular menstrual cycle, insulin resistance, excess hair growth, normally in places where most people normally have more hair growth because of the excess male hormones, fatigue, weight gain, and of course the very consistent infertility. There is no cure for PCOS, but there are treatments that can manage symptoms, and they are typically personalized to the patient based on the symptoms they are experiencing. Some of these are lifestyle changes like diet and exercise. An anti-inflammatory diet is often recommended because of the inflammation. And then some people will go on different supplement regimens that include things like berberine or minoxidil, things like that. And then also hormonal birth control, but a lot of women do not want to go on hormonal birth control because of the long list of side effects. And then fertility treatments for the infertility. So PCOS is considered an a pro-inflammatory state, and this is because of the elevated persistent levels of side effects. And several of these have been studied and proven to be elevated, and those are TNF alpha, IL-18, IL-6, and CP1, and CRP. And IL-10 has actually been proven to be reduced in PCOS patients, and IL-10 is an anti-inflammatory. So you have an increase in pro-inflammatory cytokines and a decrease in anti-inflammatory cytokines. And these are very important in helping to regulate the processes of the ovarian cycle, which I will get into on the next slide. But they basically help to regulate follicle development, oocyte maturation, steroidogenesis, and corporal inflammation. So the ovarian cycle has 3 phases. The first is the follicular phase, and this is where the cytokines help to regulate follicle structure, regulate follicle development, and oocyte maturation. And this is important because at the beginning of the follicular phase, there are multiple oocytes that are, or follicles that are being developed. And at a certain point, only one continues to maturation and ovulation. And the cytokines basically regulate this by inhibiting cell growth of the extra follicles. And so the follicles go through follicular atresia, which is where they stop growing, and then apoptosis, which is cell death. If there is a dysregulation or an elevation in cytokines, it's going to cause a dysregulation in the development of these follicles. And so after the follicular phase, you have ovulation, which is where the oocyte is released from the ovary, and the cytokines help to promote a timely rupture of the follicles by helping to break down the follicle wall. And then after ovulation, you have the luteal phase, which is where the corpus luteum is formed from the ruptured follicle that is left behind. And this basically just releases hormones in preparation for the implantation of a fertilized egg. So as I said before, anovulatory infertility is very associated with PCOS, and actually 80% of these cases are from patients with PCOS. And the first line of treatment is typically superovulation drugs like pumpkin citrate, also known as Clomid, or gonadotropins. And these just mimic the process of the ovarian cycle by stimulating the ovaries to produce multiple mature eggs or oocytes in one cycle. This is often used in majority of fertility treatments. So in IVF, for example, a patient will take a superovulation drug to produce multiple oocytes, and then once they're released, they will be harvested and turned into embryos, which can later be implanted. But for 20 to 40% of PCOS patients, they do not respond to these treatments, and the mechanism behind this resistance is unknown. So the purpose of this study will determine whether elevated inflammatory cytokines contribute to ovulation induction failure in PCOS by correlating systemic cytokine levels, ovarian gene expression, follicle structure, and oocyte release after superovulation. And as I've shown, there is already supporting evidence that PCOS is a chronic inflammatory condition, and that the cytokines play an important role in ovulation and the process of the ovarian cycle. But the gap is that there's no existing studies that have ever examined whether the inflammation directly causes or could predict ovulation induction failure in PCOS. So for my N1, I will be identifying a correlation between cytokine inflammation levels and the number of oocytes released in mice with dehydroepiandrosterone or DHEA-induced PCOS after induced ovulation. And my hypothesis is that DHEA-induced PCOS mice with higher cytokine levels will release fewer oocytes compared to the control group. So the first part of my methods is that we will obtain the mice. There will be about 40 female mice, and they'll be 6 to 8 weeks old because that is when they reach when the reproductive system reaches maturity. And then they will be split into 2 groups at random, and a control and experimental group. And then PCOS will be induced. So the control group will get sesame oil, they'll get 0.1 milliliters per 100 grams of body weight, and the DHEA will be induced into an experimental group, and you'll get 6 milligrams per 100 grams of body weight. And this injection will take place once a day into the lower left or right quadrant of their abdomen, and it will happen for 21 consecutive days. So then after PCOS is induced, you will measure the baseline cytokine levels just to get an idea of where their inflammation levels are at pre-superovulation. So we'll do this by using a blood sample and running an immunoassay panel, which will measure IL-10, NTP1, TNF-alpha,
IL-6 and IL-1 beta. And then after we got those baseline measurements, we will then start the superovulation process. So they will get 0.5 milligrams of PMSG or pregnant mare serum gonadotropin, and then we'll wait 46 to 48 hours to give them another injection of 0.5 milligrams of human chorionic gonadotropin or HCG. And once superovulation has taken place, we will then get the post-ovulation cytokine levels, which will be done the same way as before using the same immunoassay panel. And then the mice will be euthanized in a humane way using carbon dioxide, and their ovaries and their oviducts will be removed. And then to count the oocytes, we will remove them from the oviducts, and then once they're all counted, we will label specifically the experimental PCOS mice as either responders or non-responders. The responders will raise more than 10 oocytes, so that just means that the superovulation treatment did work for them, and the ones that released less than 8 oocytes will be non-responders, so the superovulation treatment didn't work for them. Our expected outcomes at the end of game 1, we're expecting to see sort of like a gradient, so the control group will have the lowest inflammation levels and the highest oocyte count, while the non-responders will have the highest inflammation levels and the lowest oocyte count, and the responders will fall somewhere in the middle. And so for A2, I will determine the impact of PCOS and inflammatory cytokines on structures inside the ovary, as well as local cytokine G expression in the ovary. And the hypothesis for this game is that PTA-induced PCOS mice with higher cytokine levels will have different follicle development, corporal lutea formation, and higher local cytokine G expression when compared to the control group. So the methods for AIM2 are pretty simple. We'll take a histological analysis of the ovaries from the mice from AIM1, and we will examine the follicle structure and corporal lutea formation, which will just help us to see if there are any changes in development between the groups, and if there are any other structural changes that inflammation could be causing. And then we will take a qPCR test, which is a quantitative polymerase chain reaction, which will measure the local cytokine gene expression in the ovary. And our AIM2 expected outcomes are that the PCOS mice with higher cytokine levels will have different follicle development and structure, as well as fewer corporal lutea, and increased expression of pro-inflammatory genes from the qPCR test. And this is an example of what a qPCR analysis looks like. So the implications of this study would be that it identifies inflammation as a potential driver of ovulation induction failure in PCOS, and supports the development of biomarkers to predict which PCOS patients will respond to fertility treatments, and it opens the door for personalized reproductive care in the future. And for future studies, I would like to test anti-inflammatory treatments to restore the ovulatory response, and also conduct human clinical studies to validate that cytokine signatures are predictors of fertility treatment success. I would like to thank my friends and family, Dr. Adams and Dr. Salvatore, for helping me through this process, Southeastern University for giving me my last 4 years, and God for everything.
In AIM1, you chose to use the DHEA to induce PCOS. Is there a specific reason why that one?
So I chose that because I had read another study where they had used multiple different methods of inducing PCOS in the mice. It was DHEA or letrozole, and in the DHEA group, they found that it leaned more towards a reproductive subtype of PCOS. So the mice kind of mimicked the symptoms of PCOS patients more in a reproductive way.
How did they induce? Like, how does that chemical induce PCOS?
So I actually didn't really look into that. I just know that I've read multiple studies, and it seems that a lot of them use DHEA to induce PCOS. There are ways after inducing PCOS into the mouse to check and make sure that they have it, and that is something I thought about
added like maybe a few extra mice to test and make sure that it has been reduced correctly.
I know, so cytokines are really important in the immune system. Do PCOS cytokines, do they affect the immune response?
So another thing that I also should have added to my future studies is that looking at the amount of like white blood cells and such that are also in the ovary for that immune response, because an increase in cytokines is also going to kind of cause an increase in like the immune cell response. So yes, I didn't add that, but that was one of the future studies. So, yeah.
See that person by the tree, then I could put it on the
Okay, I'd like to introduce our next speaker, Rocco Vargas. He's majoring in biology with a concentration in medical sciences, also a minor in chemistry. He was born in Hawaii, but now resides in Orlando, Florida. He's been at SEU for 4 years. While at SEU, he's been on the SEU wrestling team, TA for Intro to Science and Technology, and Organic Chemistry Lab, as well as a lab manager assistant. He participated in SMDA, ACS, PHS Summer Clubs, and serves as the outreach officer for the Flora Club. Upon graduation, Rocco is decided to pursue medical school and serve with the Air Force military.
Good afternoon. My name is Rocco Vargas. It's a pleasure to present my capstone and research I've been conducting here at Southeastern University the past year on the synthetic and computational evaluation of remodified effervirus analogs for sustainable HIV therapy. So for this presentation today, I want to dive in and give you a little quick snapshot of how we conduct drug discovery research here at Southeastern University. So first, let's see what exactly is HIV, or human immunodeficiency virus. So this is a virus that basically attacks and affects our human immune cells, specifically T cells that help coordinate our immune system. And how does this do it? So biologically, it is transmitted by bodily fluids such as blood, semen, vaginal fluids, and even breast milk. And so essentially, it targets the T cells that helps coordinate our immune system and attaches to a CD4 receptor. And it undergoes a specific conformational change where it helps bind and fuses inside the immune cell. So naturally in our human immune cells, we have double-stranded DNA, right? However, what makes this virus special is it carries single-stranded RNA. And essentially, it releases its contents and undergoes a specific important step called reverse transcription that transcribes it from RNA to DNA, where it then enters into the nucleus and integrates into the human genome and becomes a factor to keep replicating this virus and spreads throughout the body. So as of 2024, globally, there are over 40 million people living with HIV. However, with improved therapeutics, this number of HIV infections and HIV-related deaths has decreased within the past decade or so. However, there's still no cure for this disease. There are only treatments. And what I want to speak about today is effervirus. So effervirus is a non-nucleoside reverse transcriptase inhibitor, inhibiting that essential step of reverse transcription. It was first introduced as a first-generation drug in 1998, and however, there are still many complications and limitations to this drug. The main one is the cost. So about 2/3 of HIV patients live in sub-Saharan Africa, where access to healthcare and these therapeutics is difficult. So essentially, in the US, it costs about $11,000 to $20,000 for this drug. However, with generics, this cost is lower. In addition, there are various severe neuropsychiatric side effects, such as vivid dreams, anxiety, depression, and even suicidal thoughts. That makes patient compliance on this drug very difficult. So essentially, how does this drug work? So it targets the reverse transcriptase enzyme, and instead of binding competitively to where RNA will bind to, right here in the middle, and polymerize to DNA, it essentially binds to an allosteric site outside of the competitive binding site and binds non-competitively and distorts and changes the conformation or the shape of the enzyme prevents the further polymerization. Right, so I was able to model this for you in a crystal structure. So the green is the enzyme or the protein, and we can see, when we zoom in, exactly the drug, our antivirus, and how it interacts within the molecule. So we could investigate and look at the specific interactions that makes it very effective within the enzyme. So we were able to see that this specific side chain, the cyclopropylacetylene in this red box, was essential in acting kind of as like a key to a lock within this protein and inhibit the transcription. So in previous literature research, they have investigated modifying this side chain and have seen various biological effects in inhibiting reverse transcriptase. So the question I wanted to pose was, can we create and screen a diverse library of possible compounds for our drug? So for my specific A1, I'll use an AI-assisted model to help design and come up with more than 100 different analogs or possible structures to inhibit reverse transcriptase. So my hypothesis is that I would systemically generate different antivirus analogs and target modifications that exhibit superior binding affinity and novel interactions within the enzyme. My null hypothesis would be that I would not generate any significant binding affinity analogs. So for my method, again, I want to emphasize that I am, again, modifying the specific part of the drug that's essential for its binding affinity. So what I used was a peer-reviewed GPT as a tool to help me learn how to code and modify this specific part of the drug systemically. And this Can-Refine GPT is being used today in pharmaceutical industry for drug research and drug development. So I was able to screen multiple, even hundreds of different types of HIV drug therapeutics and looking at structures and come up with different compounds. And what it does, it creates a table for me. So here's a sample of some of our results. And on the left is a kind of shorthand notation of those chemical structures. On the right is our chemical names. And then throughout the study, I'll be using a control of the original antivirus drug. And so here's a sample list of all my structures that I was able to come up with using that AI-assisted model. And so I was able to retrieve the protein or the enzyme from the online database, and then use a specific software called AutoDocVina that helps me load the protein or the enzyme into it, and I could map out and see exactly where the drug binds to and see the specific coordinates and dock my different analogs in it. And so once I dock it in there, I could put at different conformations or orientations or poses and see which has the best binding affinity and specific interactions that makes it most effective. So for my results for A1, I was able to come up with a list of structures or analogs that has the best binding affinity compared to our control group. And so here's another sample of exactly how they look like. So you can see different variations of different size structures, very polar, non-polar, and different bicyclic structures, which is very interesting. And so for specific AIM2, I'll use a computational analysis using Swiss ADME to predict blood-brain barrier penetration to kind of address those neuropsychiatric side effects that we don't want. So for my hypothesis, the Swiss ADME prediction modeling would identify analogs that reduce the predicted CNS exposure compared to our control drug effervescence. My null hypothesis, it would be that I would not identify any significant analogs with reduced BBB penetration. So for my methods, I'll use Swiss ADME again. And what it does is when I get my list of structures or analogs, it loads it into this graph, or basically maps it out into like a boiled egg, right? So anything in the yellow models that it will be predicted to passively cross the blood-brain barrier. So what I see is our control drug crosses it readily, and which causes those neuropsychiatric side effects. So we want our drug to be more on this edge or more in the white, so it doesn't cause those side effects that we don't want. And so here are the results of our lead compounds based on binding affinity and side effects. See these top four. So on the top left are cyclopentacetylene, cyclohexylacetylene, and our 2 bicyclic structures. And so for specific aim 3 is to develop and optimize the scale of synthesis for our top results and our lead compounds. For my hypothesis, to establish a scalable protocol for our lead analogs achieving yields greater than 70%. For my null hypothesis, it will not achieve anything greater than 7% yields. So for my methods, I'll use previous research done by Dr. Salvatore on optimizing the synthesis for our cyclopropylacetylene, our original. Start from cyclopropyl ketone and do a dichlorination followed by a dehydrogenation reaction. However, as we learn in organic chemistry, or as you guys get there, you experience high ring strain with these cyclopropyl groups. So it's very difficult to work with these specific structures, as high ring strain means a lot of ring opening byproducts. And so we had to optimize the conditions, keep it at very negative temperatures to control that ring size. And then we can use the same synthesis for our larger hydrocarbon rings and minimize that ring strain. So you can use that same optimized synthesis as well as develop and investigate another synthesis where we start from hydrocarbon chain. So instead of worrying about containing that cyclopropyl group and that ring strain, we can start off with the hydrocarbon chain and do an intramolecular ring cyclization using cesium bases. And then for our bicyclic structures, we have a proposed bicyclic synthesis, multi-step synthesis, thanks to Dr. Peter Whiff at University of Pittsburgh, who came here on a visit for our seminar and was kind enough to help us develop and propose a synthesis for the structure. So this has never been synthesized before, so we're very interested in working with this. Right now, we are currently in the 3rd step of this synthesis, and I actually have the flash burning upstairs, so sometime today or tomorrow, I have to work with that. And then again, I was able to dock and see the specific interactions of that bicyclic structure that we're interested in. And again, when we zoom in, you can see different interactions with those bicyclic butyl groups within the surrounding amino acid residues. And so for future studies, we want to optimize and scale our synthesis, of course, and expand our computational library of different analogs and investigate various different structures, as well as move on to in vitro biological validation, where we do IC90 determination to determine how much concentration is needed to inhibit 90% of the enzyme, as well as in vivo studies in rodent models to investigate ADME profiles such as absorption, metabolism, excretion, and toxicity. And so the significance of this research, this capstone, is to show that every tool that I used, all the AI models, they were all the softwares were all free and accessible, open access, so anyone could do it. And so all the skills I learned is fully reproducible in industry level, as well as the skills. So here are my references. Acknowledgements, I'd like to thank, of course, the Department of Natural Health Sciences and Department of Chemistry at USF that was able to help provide resources, as well as God, of course. I would not be here without him. And my family, right here in the front row, and my grandparents who came from Hawaii. And then, of course, some of my teammates and roommates, friends, and the rest of the team for their support. Of course, again, my research teammates, we got Luke, Kaya, Philip, Jovin, Kobe, Owen, Ben, Dylan, Josh, Elena, and Noah. Of course, I'd like to emphasize my thanks to the Natural Health Sciences for providing help and being able to provide me the Harvard Next Travel Grant for sending me to Harvard Medical School to present this research. And of course, you have Joe in here. Thank you. Thank you.
So, with specifically HIV meds, I'm not involved in this, but they use a lot of combination cocktails And so would combining another medication affect the binding of the EFB?
Yes, good question. So, yeah, so a lot of HIV therapies are used in combination. So efavirenz is used with integrase and protease inhibitors called AAA. And actions with different drugs using different targets. That would be interesting to look at. But from my guess, my knowledge, I would think it wouldn't have much interaction as it targets different parts of the cell cycle.
Quiero estar caminando.
Now, how does that work specifically? Is it pulling from database of known sites because you know what you're doing, or does it come from
Yeah, good question. So again, the AI model is used as a tool, right? So it's used in current industry pharmaceutical research for drug discovery, and what it does is they train it to kind of based off online databases of known drugs and known structures and known chemical compounds, and basically includes that information, and was able to help me code and provide structures.
So would the use of this tool, would it help to make the drugs or the analogs more cost effective than efavirenz?
That's a good question. So the cost effective, are you talking about the use of the tool or for the synthesis of those compounds?
I guess a little bit of both.
Okay, so yes, because it is a free tool that I use, and it was kind of more of just a predictor, right? So this is just to help screen and give us some lead compounds, and then if we wanted to do more advanced research, of course, put more money into it and develop those AI tools. And for the synthetic point of view, for sure, for the bicyclic structures, because they were high strain, they are harder to work with, more multi-step, so this makes it more expensive. It was more just of a organic or medicinal chemistry point of view of, can we make this and maybe would it be better? You know?
If the bicyclic structures have range strain, why would you use them?
Yeah, good question. So these bicyclic structures, specifically with the acetylene or the triple bond, those have been, to my knowledge, my research, those haven't been made or synthesized, right? So we're very interested as a chemistry point of view is, can we make this, and so is this possible? And with the computational modeling, it was able to show, because specifically this one, it kind of looks like the original cyclopropyl with the triangle, right? But because it kind of has like those two cyclopropyl proteins back to back, I think it provides more surface area, more contact, and more interactions with the surrounding amino acid residues.
When you did your modeling studies, did you do this modeling studies in a gas phase or an aqueous solution?
Yeah, good question. So for my free software and stuff, I was used in gas phase. If you want to do it in solution or solvents, you would have to use more advanced software and have more resources like supercomputers, right? And so those would take weeks to process. Where I did this in my desktop in my dorm.
That concludes all of our capstones for today. And then I think at 1 o'clock, we're having the research talks in this room.
Correct, in this room. So we'll begin those at 1. So if you want to take a break, go to the restroom, grab a drink, whatever you like, and come back at 1, that would be great.
Thank you.
Yeah, they do the research talks.
Are you staying for the research talks?
How long are they gonna be?
You don't have to stay for them. Let's just leave it running. Let me check my battery. Yeah, I got plenty of battery. Let's just leave it running for the research talks, and I'll sit here and get it.
Okay.
I mean, because I have to be in this room.
Like if I need to stay, I can. I just have to be gone. Like I have to leave by like.
No.
Yeah, I was
Every time I see
I know, because I had it, typed it wrong once on the, like, just fix it. I'm sorry. I'm sorry. I pinned it. And I know. That was when I changed last one. Yeah, like, I was like, oh, that's perfect, yeah, see you tomorrow. Yeah. I would say last year, but they don't We'll have to write out. We had actually locked in today. Trying to get through all of email and
Mine is third. Addison. She's in the room, the other room right there. She is, yeah. Are you doing the first two? Are you introducing the first two? Okay, I'll do the third one. I do you, and then or the third one, and then Matt does the last two. Yeah.
All right, everyone, welcome. It's been quite a crazy day, but we're glad that you stuck around. This is the last session of the day, and this is going to be our research talk. So the first students that will be presenting are students who have done a clinical research residency program that we have developed with LRH. So those will be the first two students. And then we'll have some physics research. I think this is the first time for that. So that'll be happening, being presented today. And then also some biology research presented here as well. So it should be very exciting, and I hope that you enjoy it. So let me introduce our first speaker. So Anna Church, can you all hear me okay? Okay. Anna Church is majoring in biochemistry and is from South Bend, Indiana. Go Indiana. She has been at SEU for 3 years, and while at SEU, she has TA'd for Biology 1 and 2 and Ecology, has been a side peer mentor, and a member of SCMDA, KME, and PHSO. After graduation, she plans to attend medical school. So welcome, Anna.
Hello, everyone. As Dr. McConkey said, my name is Anna Church, and today I'll be presenting the study that I designed under the guidance of the Lakeland Regional Research. So the title of my study is called The Metabolic Modulation of Alzheimer's Disease Biomarkers. To begin, I'd like to state that I have no conflict of interest to disclose. So first, a little bit of background on Alzheimer's. I'm sure we're all pretty familiar with it, but it's described as a progressive, irreversible neurodegenerative disease. It affects 1 in 9 Americans, so that's kind of over the age of 65. And it's described as it is characterized clinically as a decline in memory, cognitive function, and behavioral changes. Currently, there's no effective treatment for Alzheimer's disease, and there's several risk factors that can put somebody at a higher risk for Alzheimer's disease, and that can include smoking, reduced estrogen levels, high cortisol, obesity, inflammation, and type 2 diabetes. Cellularly, it's characterized in 2 main ways. Now, while the mechanism of neurodegeneration isn't fully known, there's 2 main ways that they characterize, and that's the extracellular deposition of beta amyloid plaques and the intracellular accumulation of these hyperphosphorylated tau proteins. That's what I'm going to be focusing on in this study. So as you can see up on the screen, that's a healthy microtubule in a neuron, and the tau proteins work to stabilize that. When you have Alzheimer's disease, these tau proteins become hyperphosphorylated, and that decreases their binding affinity to these microtubules. And then they can break off and then clump together, and they form neurofibrillary tangles. So this is a really complex pathway. I'm just going to give you the highlights that insulin resistance, when insulin binds to the receptor, it will trigger this whole pathway that's going to lead to inhibiting what's called GSK3 beta. GSK3 beta is responsible for regulating these tau proteins binding affinity. So in this pathway, when we have insulin resistance, that enzyme is chronically activated, which means that they become hyperphosphorylated and then they are unregulated and they branch off. So currently, the studies that are being performed are long-term and observational, and they don't have any short-term interventional human studies that examine the improvements of metabolic biomarkers on these plasma p-taus. Additionally, they do not assess these specific plasma p-taus because the materials that we're using to do this is relatively new. So the purpose of this study is to evaluate the effects of the changes in insulin resistance on these phosphorylated p-tau proteins following a ketogenic intervention. And this would be in specifically non-diabetic, insulin-resistant, cognitively intact adults ages 30 to 50. So that leads me to my first specific aim. First, I'd like to evaluate whether the ketogenic diet can modulate these fasting p-tau levels following that intervention. And my hypothesis is that with a decrease in insulin resistance caused by the ketogenic diet, we will see a decrease in these hyperphosphorylated tau proteins, which kind of encompasses my specific aim too, which is to evaluate the relationship. Will we see an increase in insulin sensitivity? Will we see an increase or decrease in these p-tau proteins? And the hypothesis is that it will be positively correlated. So the study design is a prospective interventional study, and it will be conducted at Lakeland Regional Health. To recruit patients, they will be identified based on predefined inclusion-exclusion criteria. And this will be through medical records and physician referral. And we would, of course, use a convenient sampling approach. So there's a lot on here. I'm going to go through it kind of quickly. But the inclusion, in order to be a participant in this study, you need to be between the ages of 30 and 50. You need to be insulin resistant, but you cannot be diabetic. So that means your hemoglobin A1C needs to be below 6.5%. You need to have a homeostatic model assessment of insulin resistance of between 1.5 and 2.5, and your fasting glucose levels need to be below 125 milligrams per deciliter. Additionally, you need to be cognitively intact as described by the Lipton assessment. Exclusion, you'll be excluded from the study if you've participated in any metabolic diet within the last year, if you've been diagnosed with Alzheimer's disease, cardiovascular disease, heart disease, kidney disease, liver disease, or any other neurodegenerative disease. Additionally, you will be excluded if you're on currently any medications that affect insulin resistance. And if women are participating in this study, currently pregnant, postmenopausal, or women with irregular menstrual cycles will be excluded. So the intervention. So patients will be assigned a group, and I'll talk about this group in a minute, and they'll follow this strict diet. Then every day at their home, they will be responsible for a ketone urinalysis test. This is just to confirm that they are in fact in ketosis. They will record that in their digital patient journal. Now, the assessment. There'll be 3 assessments. There'll be one at the baseline. This is before they begin their diet. Then there'll be another assessment at the midpoint, and a final assessment after the 4 weeks. This assessment will be the same throughout. This will include the homeostatic model assessment of insulin resistance. This will include the p-tau specific assays, and we will record their menstrual cycle if the female participants. So the 2 groups. So group 1 is going to be our interventional group. This will receive the isocaloric ketogenic diet. Isocaloric meaning they're going to receive a consistent caloric intake, so that would be controlled for weight loss. Group 2 will also receive an isocaloric diet, but it will not be a ketogenic diet. Now, I'm sure we're all wondering, what is a ketogenic diet? The NIH defines this as 60 to 70% of our calories that we would eat on a ketogenic diet would be from fats. Okay. And then 25 to 30% would be from protein, and then a small fraction would be carbohydrates. And this is to induce what's called ketosis, which is a metabolic state where your body shifts from using glucose to using fats. And this is done when insulin levels are low. This stimulates the breakdown of fats, which then is converted in the liver to ketones, which can create an alternative energy source. So the study design. First, we obtained IRB approval. Then we would collect baseline assessments. They would begin their intervention. Then we collect midpoint assessments. They would continue their intervention all the way to 4 weeks, and we collect their final assessment. So statistical analysis. We would collect continuous numerical values for this, but I'd be working in collaboration with a biostatistician to perform the appropriate tests. Additionally, we will account for menstrual phase statistically. So there's a couple safety considerations with the ketogenic diet, and this can include short-term side effects during the adaptation phase. And this is because there's something they call it the ketogenic flu, and patients may experience vomiting, diarrhea, and this is just as you adapt to switching from glucose to ketosis. And it can be managed most of the time through adequate hydration. Now, in rare cases, more severe complications may occur, in which case those patients will be removed from the study. Now, ethical considerations, obviously IRB approval will be obtained. Patients must be able to provide informed consent, and their participation will be voluntary. We will be obligated to protect their data for long-term storage, and we will be obligated to report any adverse events. Additionally, any major study findings will be communicated to participants. So our anticipated results. We anticipate that with a decrease in insulin resistance as caused by that ketogenic diet, we will see a decrease in these biomarkers in p-tau as measured by our p-tau axis. And this would imply that metabolic intervention can modulate plasma p-tau levels, and that the ketogenic diet may be an effective means for doing that, and that insulin resistance may be that upstream biomarker, that upstream driver for the development of Alzheimer's disease, or one of them. And this is very significant because this potentially identifies the prevention of Alzheimer's disease. These are my acknowledgements. Happy to
Out of all the different correlations that there are with Alzheimer's disease, why is this specific?
So, my grandmother had Alzheimer's disease, and when she had that, I was one of her caregivers. And as I was doing that, I was reading several books about Alzheimer's disease, and one of them was talking about the ketogenic diet as a potential treatment for Alzheimer's disease, and saying that Alzheimer's disease is type 3 diabetes. And so I was like, I wonder if that's true. So I looked into it, and I couldn't find any literature that was clearly defining that it was. It just was one of the potential causes. And there have been no official studies that I could find that showed monitoring these specific biomarkers with the ketogenic diet or the ketogenic diet in general. Yeah, other questions?
Studies 30 to 50-year-olds, where typically Alzheimer's is 65 and older. Why this stage?
So that's because we want to identify an upstream before the clinical onset of symptoms. So often they'll start experiencing like minor cognitive decline once you get up beyond 50. So we emphasize that between 30 and 50, we will see the buildup of these tau proteins in a measurable, meaningful way, but not to the extent of
So these patients have the symptoms early onset.
no, they would not.
Is a 4-week diet a typical amount of time for the keto diet?
So generally, from what I could find, they suggest being on it long term, but you will start to see changes in it that could be measured within a 4-week period.
Okay, so statistically or significant changes would happen after the 4-week diet.
Potentially, yes, yes. It's a range of things.
Any other questions? And we would test the liver.
Yes, so it'd be the plasma levels, so it would be a blood draw.
All right, thank you, Anna.
All right, so,
I didn't write it down. I just have the presenter saying.
give y'all time to get rearranged here. There's some empty
Yeah,
seats
Anna,
down
sure.
here. If you all want seats, there's 3 down here in the room. Yeah, I was like. Okay, we're gonna go ahead and move on to our second speaker. I can get everyone's attention. You want to stay on time? Thank you. So our second speaker is also a student who conducted clinical research at LRH. And it is Milianis Garcia, and she is majoring in biology with a concentration in medical sciences. She's from Puerto Rico and has been at SEU for 4 years. While at SEU, she has been a member of CNHS, SCNDA, and served as an SEU clinic volunteer. Upon graduation, she plans to attend Lake Erie College of Osteopathic Medicine in Bradenton starting this summer. So super exciting. Yeah, go for it. You want to point
Hi again. So my name is Milianis Garcia. I'll be presenting the research that I was able to put together with the LRH clinical research team. So the title of my research is Omega-3 and choline supplementation as a neuroprotective strategy in infants with critical congenital heart disease following neonatal cardiac surgery. Right, so first of all, there are no conflicts of interest in this study. So for a little bit of the background, congenital heart disease, or CHD, is a structural abnormality of the heart. It's the most common birth defect. We see it in about 8 in every 1,000 babies each year. And so neonatal cardiac surgery results in increased risk for brain injury, especially white matter injury. New white matter injury occurred in 44% of newborns after they had CHD surgery. So we're focusing on babies who had critical CHD. Critical means that they needed to get surgery for this disease that they were born with. And so we also see neurodevelopmental disability, which is the most common side effect after CHD surgery in about 30 to 50% of infants. So this is a really significant number. And in the pictures here, you can just see on the left is a healthy brain. And then on the right, I want to point out that is a neonatal hypoxic ischemic brain injury. So you see there's hypomyelination, neurotoxicity because of the inflammation. So we're doing, you guys see that okay? It cut off a little bit, but so omega-3 and choline, why would we use this as a supplement? So omega-3 fatty acids reduce inflammation. So as you saw in the other slide, we see that inflammation is one of the risks. And then choline is also necessary for cell membrane and also myelination. We also see that it's very important in early brain development as far as neurotransmitter synthesis. So studies have shown that both of these are generally safe for neonates. So the gaps in knowledge here, we do not have any treatment that prevents white matter injury at this time. And we also have limited evidence on reducing long-term neurodevelopmental delays, and also limited research on nutritional neuroprotection, as well as we haven't studied the effects of omega-3 and choline supplementation on neonates post-operation. So the purpose of the study is to evaluate whether or not early post-surgical omega-3 and choline supplementation improve white matter maturation, neurodevelopmental outcomes, and whether it is safe and feasible in infants with critical CHD. So my specific aim 1, so we aim to assess the safety and feasibility of early post-surgical omega-3 and choline supplementation in infants with critical CHD. And the hypothesis is that the supplementation will be well tolerated and safe, with no significant increase in adverse events or feeding intolerance. Our specific aim 2 is to evaluate the effects of early post-surgical omega-3 and choline supplementation on white matter maturation in infants with critical CHD. We hypothesize here that infants will show improved white matter maturation on diffusion MRI after the supplementation. So in specific aim 3, we're hoping to evaluate the effects of early post-surgical omega-3 and choline supplementation on the neurodevelopmental outcomes in infants with critical CHD. And again, here we hypothesize that the infants will demonstrate neurodevelopmental scores closer to age-based norms. So we'll be doing this. This is a study that's prospective, longitudinal, pre-post interventional pilot study. We'll be doing it at LRH, specifically in the NICU. And then we'll be using neonates diagnosed with critical CHD requiring surgery within the first month of life, and we'll be doing about 20 neonates. So for our methodology, like I said, this is a pilot study on neonates diagnosed with critical CHD. We're evaluating the early post-surgical omega-3 and choline supplementation. It is a once daily dose, 24 months long, and again, 20 participants. So for my inclusion and exclusion, like I said, it's neonates diagnosed with critical congenital heart disease. Their ongoing surgery in the first month. They have to be hemodynamically stable. That means their blood pressure is okay, their oxygen is okay, their heart rate's okay. And then they have to be eligible for enteral feeding. So they have to be able to drink their milk and keep everything down. And of course, we have to have informed consent. Any babies with major genetic syndromes, pre-existing significant neurological injuries, severe prematurity, contraindications to MRI, or an inability to tolerate feedings, or hemodynamically unstable, all of those babies will be excluded from the study. The intervention, like I said, is omega-3 and choline supplementation. So the choline supplementation changes every 6 months. From 0 to 6, we'll do 125 milligrams a day. 6 to 12 months, 150 milligrams a day. 12 to 24 months, 200 milligrams a day. Omega-3, we'll be doing 500 milligrams of omega-3 once daily for all 24 months. So again, we're doing this enteral route. So through their feedings, supplements are not FDA approved, but studies have been done that deems this as a safe intervention for infants. So to measure our outcomes for AIM-1, again, I'll remind you, AIM-1 is to assess safety and feasibility. So we'll be checking for adverse events, feeding intolerance, gastrointestinal intolerance, ability to complete feedings. We'll be using a caregiver log to capture daily supplementation adherence and any issues throughout the study. We're also just evaluating the ease and also, again, just if it's safe and feasible. For our outcome 2, like I said, for specific AIM-2, we're checking the white matter. So we're using a diffusion magnetic resonance imaging, or an MRI. Every 6 months post-supplementation, we'll have a baseline, and then every 6 months, they will get MRI. As you can see on the top picture right there, that is a brain with hypoxic injury. On the bottom, that is age-specific norms. So we're hoping that as the months go by, we can see something closer to the second picture instead of the first picture. So for measuring outcomes for AIM-3, again, for this one, we're measuring neurodevelopmental outcomes. So we'll be using the Bayley scales of infant and toddler development. The most up-to-date is Bayley 4. We'll also be doing this every 6 months. We're trying to make this easier on the parent, just bringing them every 6 months. Here are some examples of the scores, and then we're going to compare the scores post-supplementation to age-based norms. So the study procedures, we're going to identify the eligible neonates, obtain informed consent. We're going to collect baseline data. We're going to, then the baby will go on to have surgery. Then we'll monitor until they're clinically stable. That takes about 24 to 72 hours. And then we'll be able to initiate choline and omega-3 supplementation. And like I said, follow-ups will be done every 6 months. The safety and adherence will be monitored throughout the 24 months. So here's a quick timeline. We kind of already talked about this, so we'll go through it quickly. So the baby will be born and diagnosed with critical CHD. Then they'll be assessed. We'll do baseline. Then they'll go into cardiac surgery. We'll stabilize them and initiate the supplementation. Then we're going to monitor the safety and caregiver diary throughout the 24 months. We'll do a 6, 12, 18, and 24-month follow-up to do the MRI and Bayley 4 assessment. And then at the end of the study, we're going to do a data analysis and outcome evaluation. For our analysis, we're going to work with a biostatistician to just perform all of the analyses in the study. And then the expected results, we expect that supplementation will be well tolerated, no unexpected safety concerns. The MRI are expected to show gradual improvement in white matter maturation. Neurodevelopmental outcomes are expected to be closer to age-based norms compared to the historical expectations for infants that went through CHD surgery. And then the results may suggest a potential neuroprotective signal of this supplement supporting further larger scale studies. So for safety and ethical considerations, of course, neonates are a vulnerable population, so it will require additional IRB review. And then we will, of course, obtain parental informed consent. The study will involve minimal additional risk, and supplement will begin only after the infant is stable. So study significance, this addresses a critical gap in neuroprotective strategies for infants with critical CHD. Findings will provide safety and feasibility data for omega-3 mucosan supplementation, and then the results may identify a potential nutritional strategy to support white matter maturation and neurodevelopment. So for our further studies, I would like to do larger randomized controlled trials to confirm the efficacy of the supplement and also make sure that the dosage is right, the timing, and the duration of the study. And then long-term follow-up is needed to assess how the child grows up and how that goes into childhood. So here are my acknowledgements, my references. Thank you. Any questions?
What is the basis of choosing your dosage in both of those supplements?
Yeah, that's a great question. So I was doing research of seeing how much omega-3 can be given, how much should it be given. For the first 2 years, there's a 500 milligram that they want. The limit is 700, but if we're gonna supplement, the best dosage that I could find was 500 milligrams, where it wouldn't pose any adverse events, but also show an improvement. As well as choline, you need more as they're growing in development in their toddler years. So that's why it goes up a little bit more. With babies, you just have to be careful to not give them too much. So that's why I chose to do a little bit less. And again, I did research to kind of see what is okay, what is necessary, and what doctors are doing if they are doing this.
Follow-up. Has there been a combination of that is already established? Because you have 500 for your choline and then 125 with the omega-3. That's kind of a lot for a little 28 C.
So it hasn't been established as a like together, but we I did make sure that that was a safe intervention for the neonates. But that might be a really good idea for future study.
Other questions?
Well, what type of adverse effects?
Oh, adverse events, we're making sure that their blood pressure doesn't drop, that they're not having extreme diarrhea that leads into dehydration. So basically, we're just checking vital signs and any GI issues.
One more question? Yes.
How would the results of this study would impact your practice? Because we know that you dreamed to be a pediatrician since you were a kid.
Thank you, Mom. So I've been working at LRH in the pediatric unit for about 3 years now, and I've seen lots of kids get heart surgery, and then that's not where it ends. Like, we have so many worries about their brain and neurodevelopment as they grow, and so that will definitely affect how I treat my patients. That it's not just getting surgery and letting them go home. It's really trying to get them into a better childhood overall. Thank you so much.
Tiene que entrar por comida lenta. Ya se acabó. La que nos preguntó. No se fue ahora mismo. No, pero estaba sentada ahí.
All right, our next speaker today is going to be Addison. Addison did a biology research project under my supervision this semester. Addison is majoring in biology with a concentration in veterinary medicine. She's from Tampa, Florida, and has been at SU for 3 years. While here, she has served as the vice president of the Christian Veterinary Fellowship, or Vet Club on campus. And after graduation, she plans to attend graduate school to earn her doctorate in veterinary medicine.
I'm Addison, and today I will be presenting my project titled "A Preliminary Comparison of Biodiversity on Ocean Flood and Live Rock from Australia and Florida." Basically, this study kind of focuses on the origin of live rock and whether it has an effect geologically on the overall ecosystem health in saltwater aquariums. Specifically, we wanted to understand if the geographic location had any impact on survival and the amount of biodiversity. This is important because the aquarium hobby is a multi-billion-dollar industry, but it also has a direct implication on reef conservation. So first, I'll start with our introduction hypothesis, then I'll talk about our methods. Most of our time will be spent in the results and discussion, and then I'll go into future plans. So to start, what is live rock? Live rock is essentially limestone that's been naturally colonized by marine organisms. That means that everything from like microscopic to macroscopic organisms like bacteria, algae, and small invertebrates can colonize this rock. The reason it's important is because it actually acts as a natural biological filtration system. So instead of it just being like pretty in a tank, it actually drives nitrogen cycling and actually acts as like a home for all these creatures. The collection methods and where it's from matters a lot. When we studied, we found this out more. But for Australian rock, it's collected directly from the natural reef, meaning that it's taken off of the reef, which could possibly have some negative effects to the reef. It does require a permit, but still it does kind of raise some ethical concerns when taking this rock or this coral. For the floor rock, it's actually aquaculture, meaning it is a rock placed into the ocean for a few months to years, and the organisms naturally colonize it, and then it is sustainably harvested after colonization and submerged fully in water. Sorry. So why does this research matter? It matters because marine biodiversity varies a lot depending on region. In regions like Australia's Indo-Pacific region, they have the highest biodiversity overall marine-wise. So looking at this compared to Florida is pretty important and can help us kind of in aquarium hobbyists kind of decide where they want to source their rock from, because it can show us the effects of not only like conservation, but also how their tanks will thrive and what organisms will kind of pop up in the tanks themselves. As I said earlier, the aquarium hobby is a multi-billion-dollar industry. So the choices that these hobbyists make has like a direct implication on our reef system and overall ocean health. So, our hypothesis was that the Australian rock would have greater biodiversity due to the fact that it comes from that Indo-Pacific biodiversity hotspot. The shipping method, we kind of sort of maybe thought might have an implication because the Australian rock is shipped wrapped in damp paper towels, whereas the Florida rock is shipped fully submerged. So when the rock comes in, we thought that maybe the Florida rock might have a bit more life to start. For our methods, we set up 4 10-gallon tanks. Each came equipped, or we provided a power filter, a wave maker, and a heater to try to keep conditions as consistent as possible so that any observations observed would be from the rock itself and not from the surrounding environment. We tried to keep things controlled like temperature and salinity in order to keep things again as controlled as possible. But in tanks 1 and 4, they did have a lower light, whereas tanks 3 and 4 had a higher light. We also we controlled water flow and filtration using a Hobb filtration system. And over this experiment, it was 10 weeks long, and we did 10% weekly water changes along with algae removal from the glass. And we topped off the evaporated water about every other day using RO water. So looking at our temperature data, we can see a range from about 75 to 78 degrees Fahrenheit, which is pretty optimal for reef tanks. As you can see from our graph, about every graph was very consistent altogether. What we kind of observed is that temperature likely did not have any effect on what organisms that we observed, and any organisms that were, or any different diversity was probably not affected by the temperature at all. Talking about salinity, our range was from 1.022 to 1.03 specific gravity, and kind of the range you want your tank to be is 1.025 to 1.026 specific gravity. So we did have a bit of variation there, but what really is important is that the tanks themselves stayed consistent. And as you can see from our data, when they did kind of drop and when they did peak, they were actually at like the same kind of consistency, like the same levels. So that's really, truly important. So we also do not think that salinity had a big impact on the biodiversity of these tanks. And we also use Red Sea blue salt, which actually is collected using evaporated Red Sea water. We also wanted to quantify biodiversity as best as possible, so we used a eDNA sampling kit from Aquabiomics Testing. This allowed us to look at the microbial communities and the things we can't really see. So the advantage of this is that, like I said, it captures those organisms like eukaryotes and the bacteria and archaea, and it would essentially help us kind of quantify biodiversity. And the kit instructions were followed accordingly so that any bias was eliminated. Starting with our Australia rock, both tanks we saw pretty much no visible macrofauna. There was pretty much no life throughout the experiment visually, so things like crabs and worms and whatnot were not seen. However, we did see something called coralline algae in the tanks. However, it died off relatively early, and the tank was overwhelmed by that cyanobacteria gnats and some nuisance algae as well. But when we did look microscopically, we did see some microscopic life, so that suggested that there was still life in the tank present in those mats, just not microscopic or macroscopically. Excuse me. When we zoom in microscopically, we see more of the detail of what actually is present. So we observe some things that look like sponge-like material, as well as diatoms and even a small snail. So while the tanks look relatively inactive, there was still life present in a micro community. So it just seemed to be pretty much dominated by those opportunistic organisms like that nuisance algae. Going into the Florida rock, it kind of showed completely different results compared to the Australian tanks. From day one, there was visibly much more life in the Florida rock compared to the Australia. We observed things as such as sponges, brittle stars, bristle worms, corals, crabs, snails, anemone, and coralline algae. And like I said, that coralline algae is a sign of healthy live rock, so it was important that we saw that. Looking at the tank microscopically, we observed again diatoms, and at like a macro level, you can see that the density of the diatoms are pretty clear and pretty dense. So this is an indicator that there was microbial life in a microbial community. That video won't play, but again, this kind of just shows the macroscopic life we saw. So in figure 24, you can see a sponge. That's what those like clear things on the rock are. And then if you look at figure 26, on that rock there are some barnacles. So this kind of, again, kind of shows us that there was both microscopic and macroscopic biodiversity in these tanks. And looking microscopically into tank 4, our other Florida tank, we can see some more microscopic life. A lot of these were kind of difficult to identify, but in figure 29 and in figure 32 on the right there, that is some sponge material that we observed under the microscope. This kind of shows us that microscopically there was a highly active microbial system. And this is just like a little video of a nematode from tank 4 that suggests microbial life. And looking macroscopically at tank 4, this is the coolest part. It had the highest biodiversity overall for sure. We observed things like, again, crabs, there was a snail, and coral and whatnot. So it really represented the strongest example of a successful live rock tank. The successful rock colonization is really shown in tank 4. And I do not have access to play these, but there was a crab filter feeding in this video, and it showed the ecosystem and how the organisms play a role in these environments. And as it pertains to our eDNA results, unfortunately, we did not receive them in time. They apparently do them in batches, and we were not made aware of that. So we will be receiving our results on August 27th of this month. But we did receive these images from their website as an example of what our results hypothetically might look like. So in figure 45, this is one of their images showing us all of the different families and their kind of proportions on that graph there. And in figure 26, it shows us how specific they kind of can get. So in figure 26, it has all the anthozoa, but you can see on the far right-hand side, they actually break it down to species and genus as well. So they can get pretty far into depth using that eDNA. And into our discussion. So our findings did not support our original hypothesis that Australian rock would be more biodiverse. Actually, Florida rock showed greater biodiversity, especially in that tank 4. Instead of Australian rock giving us those results, Florida did. But when we did unpack the Australian rock, it did have a very foul odor, suggesting decay upon transport. So overall, it seems like transport conditions kind of had more of an impact in the geographic location at this point. Why tank 4 was different. One of the interesting things was that tank 4 looked immediately more biodiverse immediately after unboxing. It showed biodiversity immediately with corals and life already on it the second we took it out of the bucket that it came in. Likely the explanation is that it was submerged and that therefore kept the life sustained. And there may have been less stress during transport. But also, of course, natural variation plays a role within samples, as this one even outperformed tank 2 or other Florida tank. So it would kind of be interesting to look into that further. So in conclusion, Florida rock showed higher biodiversity. Overall, tank 4 showed the highest biodiversity. The transportation, most likely the submerged transport, played the biggest role in determining this factor. And there may have been less stress, like I said. It also highlights that geographic location alone doesn't really determine biodiversity, and that the transportation has an effect on what we find and what we see. And finally, the aquacultured and sustainably harvested reef actually performed better. So that kind of goes to show us that it's better for the success of these hobbyist tanks, as well as for conservation efforts. For future studies, what we could do is quantify biodiversity at day 0 using that eDNA sampling kit, and then repeating it again at the end of the experiment to see kind of maybe the different densities and what has popped up and maybe what has gone away. We could also use an auto top-off system for more consistent salinity, just to reduce variability, as well as using more microscopic work in order to kind of track the microbial community and see if anything dies off that we can see that way. And then it would also be interesting to study the light intensity kind of side of things, and also to do it for a more long-term ecosystem developmental study.
And then I would like to acknowledge God and Professor Hughes very much for his insight and mentorship. And then my partner Carson, who is not here unfortunately, but for her collaboration. And
Any questions for Addison?
For another future study, do you think that it would be useful to partner with an Australian university to do the same experiment but backwards? So they get the rock delivered in water tanks and then we send ours.
I think probably yes, because we send ours in water and they don't. I'm not sure if there's anywhere in Australia that does send it in water for like a repetition here, but that would definitely be a good option, yes. Oh, yes.
So, you mentioned for future studies, looking at different light levels. Did you measure at all, like what the lux was for the lights on these?
I do not know.
Thank you.
Yes.
Quick suggestion, don't trust the people in Australia, just go there yourself. Right. Oh, I won't go.
Well, anywhere in Australia, almost anything can kill you.
Well, she said we just stay away from places that things would be like.
All right,
It is, yeah.
continuing things on here, I have a couple students from the MS program here. First up, we have Miriam Fernandez. She is obtaining her Master's of Science in Medical Sciences. She is from Havana, Cuba, currently resides in Orlando, Florida, and has been at SEU for 1 year. Upon graduation, she plans to attend pharmacy school.
Hello. So, physics. Good afternoon, everyone. Like Dr. Ahmed said, my name is Miriam, and today I'll be presenting my project on my review on how physics is related in applications in medicine. So what I looked at was ultrasound. So I'll be presenting on modeling the trade-off between ultrasound frequency, image resolution, and attenuation in soft tissue. And the focus for this review project was to see how fundamental physics principles impact directly the quality and effectiveness of medical ultrasound imaging. Okay. So my primary research question that guided my review was I wanted to learn how increasing the ultrasound frequency from, let's say, 2 to 12 megahertz is going to impact axial resolution and image attenuation at depths between 2 to 10 centimeters in soft tissue. We'll get into the terms a little later into the slides, so don't worry, I will catch you up. And so what I wanted to do was not just describe this trade-off qualitatively, but actually try to model it quantitatively in order to see how significant these effects were. So I messed around with some of the equations to try to get some real values here, and I'll show you the trend. So why does this matter? Why am I talking to you about ultrasound right now? So ultrasound is one of the most widely used medical imaging techniques due to it being non-invasive, relatively inexpensive, and it doesn't use ionizing radiation, which makes it very safe. However, there is an impact on how effective it is because you need to balance two competing factors. That is resolution, which determines how clearly we're able to form an image, and then penetration. How deeply can we go into the tissue to actually get an image? So say, for example, a doctor is trying to get a superficial image of, say, like a tendon. They would want very high resolution in order to detect the smaller anatomy of a tendon. However, if they're trying to get deeper into the body, say an abdominal organ, like a liver or a kidney, then we have to opt for a wavelength that is able to travel that far in order to get our image, even if we have to sacrifice some detail. And so in order to understand this, then we need to go into the physics of it. So ultrasound is a mechanical wave that travels through tissue. So like all waves, it is defined by frequency, wavelength, and speed. Because it's a mechanical wave, it needs a medium, and so our medium being the body, our speed constant is actually constant at around a value of 1,540 meters per second. So that is the value that I referred to for the rest of my investigation as I try to figure out the physics of this. And because of this established wave property, when frequency increases, wavelength must decrease. And with this established, we see that at higher frequencies, we're actually able to resolve much smaller anatomy. So that means we're able to get a better image at higher frequencies. And so right away, physics is letting us know that we get better resolution at higher frequencies. Get used to those words, I'm gonna be saying them a lot. So for our core equations, I stumbled upon a few other equations that are physics related, but really the only ones that I really focused on to understand what I'm looking at specifically for ultrasound are going to be these four. So the first being the wave properties, which I just talked about. It's going to be the speed equals the wavelength times the frequency. Then our equation for axial resolution. So there are two types of resolution. It's going to be axial and then longitudinal. Axial refers to, I'm going to be looking at structures that follow the beam of the ultrasound. So they're all going to be parallel to the beam. Longitudinal is going to be when the beam is perpendicular to two structures that are next to each other. So I'm looking at a straight path for ultrasound. And then there's two formulas that came up for attenuation, which I'll explain what attenuation is, and I believe 2 more slides. So there's the exponential attenuation equation, which models intensity, and it lets us know that as a wave travels through tissue, it will lose energy. And then a frequency-dependent attenuation, which is more of a linear model that relates attenuation to frequency and depth. In order to understand resolution, like I mentioned, axial attenuation is going to be the formula we want to look at. And this is the one that I used in order to form my trend. So the smaller the resolution, that's going to be the better our picture quality is going to be, the better our image detail. And so let's say I plug in 2 megahertz for our frequency. Since our speed is constant, that will get us to about a value of 0.385 millimeters. And then if I plug in a higher frequency, say 12, then we get an improvement to 0.064. That's about a 6-fold improvement in our resolution. So again, physics is once again proving how higher frequencies are going to give us that nice and crisp image, so that we're able to focus on smaller structures when we're taking an ultrasound. So yeah, a smaller R value means better resolution. And then attenuation starts to become a problem at higher frequencies. So attenuation is when the wave starts traveling through the body, it's going to start losing energy through absorption, which is where the energy is going to be converted into heat, and then scattering, which is where our wave is going to be redirected in other directions other than the structure that we're trying to look at. So attenuation basically disrupts our ultrasound. And this, I used the linear approximation in order to see how that relates to frequency and depth, since that's what I'm really interested on. The intensity decay really only, I included it since it explains the energy loss, but I didn't use it for my modeling. So here in the first image is my resolution versus frequency. We can see that at shallower depths, we have a really high resolution. Sorry, this one's not depth. At lower frequencies, we have low resolution, which is further up. Remember that a smaller R value is what corresponds to better image quality. So then as we increase our frequency, our resolution decreases, which means a better picture. And then we can also see that it also starts, the trend is that, yes, there is an improvement, but it starts to level off. So at higher frequencies, the improvement is gonna get smaller and smaller. So it's not that it's, higher frequencies are gonna have a super better image. It like the effects of the improvement slowly change over time to a smaller degree. And then to look at attenuation, I modeled it after looking at 3 depths, so a shallow depth, so 2 centimeters, then 5 centimeters, and then 10. We can see that at shallower structures, we're able to not get as much attenuation, so our signal is not losing as much energy. We only get about 10 decibels of disruption at higher frequencies. But then once we get deeper into the tissue, we get a lot of attenuation. So our wave is losing a lot of energy, and that means we're losing a lot of resolution. So the deeper we go, the model that I used, we get to about 60 decibels. And every change in this model, since it's exponential, every 10 decibels is a 10-fold decrease in our resolution. So at 60 decibels, that's about a millionth decrease in our resolution. So that's how much it impacts our final image of what we're able to see. And this just summarizes what I said. So 2 megahertz, we only get about 10 decibels, and then that's wrong, we'll overlook that. So then we arrive at what I finally learned about ultrasound. There is a clear trade-off. It doesn't matter, rephrase that. At higher frequencies, we're gonna get a high resolution. However, it won't work the deeper we go into the tissue. So we can't try to get better resolution the deeper we go. In the result, at low frequencies, we're able to get to the deeper organs. However, we are gonna have to give up some image detail. And now the thing that a lot of the papers that I looked at really tried to drive is that this is not a technology problem. We are not lagging in our advancement for ultrasound. This is just fundamentally how the wave physics works. So there's really, as of right now, unless somebody changes physics, nothing you can do to kind of try to improve resolution at the deeper level. You're gonna have to give up that crispiness of the image. So real world applications. So then this changes how we use it in the sense that the higher frequencies are gonna only be applied when we wanna look at the superficial structures. So things like blood vessels, a thyroid ultrasound, or like a tendon, which is the example that I gave earlier. And then those lower frequencies are gonna be for when we wanna get into the abdominal organs like a kidney or a liver. However, I just said that it's not a technology issue, but there has been a kind of like a workaround. So they do make multi-frequency probes. So instead of having to change equipment every time you want to run a different test, if you're doing multiple things at once, they do make these multi-frequency probes that do have like an automation feature. And so they'll cycle from high frequency to low frequency, depending on what kind of test you're trying to run, what kind of structure you're wanting to look into. And then they'll do that automatically and also manually. So let's say you're trying to get a blood vessel and then you're like, actually, let's go look at your liver. And so you'll go change it and you won't have to change equipment. What I did end up modeling with what I learned does have some limitations, assuming a constant attenuation coefficient, which this would change based on tissue type. And so I just chose a standard one for soft tissue. And then I also ignore other effects like reflection, noise, and equipment effects. So I didn't look at specific like models of ultrasound or anything. I'm just like trying to model the standard of how wave physics works. So what did I learn? Physics has a lot of applications in medicine, and specifically in this one, it basically decides how we're able to use this equipment. And again, not a technology issue, just fundamentally how physics works. And in the end, we arrived at higher frequency, my initial question, right? How does increasing frequency go to 12? Higher frequencies do improve our resolution, but only at shallow depth. We can't get high resolution images into deeper tissue. And so this, we should optimize the frequency depending on what we want to look at, like I explained, and understanding this balance, this balance helps design ultrasound systems, and how to use them effectively. Thank you. Okay, that was a lot. What do y'all got?
Oh, where did you get the chart and the data from? Like, where's the number came from?
So this is what the typical chart for modeling resolution versus frequency looks like. I plugged in my own numbers using this formula. So I charted for using a frequency of 2, 5, and then 8 and 12. So I used my own numbers to show the trend, since that's the only thing I'm really trying to do, is show you how you improve resolution the higher you go, and how it like plateaus off the further you go. So I made them. Same thing with this one. I just picked 3 depths. So I looked at my minimum of 2 centimeters, then 10, and then I chose one in the middle just to show like how it actually looks like on the figure.
Did you see if there were any other types of waves that produce better actual resolution or attenuation data?
I did not. I only looked at papers that talked about ultrasound like mechanical waves, and specifically the ones that use axial resolution. So that's the only thing I looked at. All right, thank you.
No, I really can't.
All right, and so I believe this is last for today, but definitely not least. We have Nina Nayawara, pretty good, is obtaining her master's in Master's of Science in Medical Science as well. She is from Poinciana, Florida, and has been at SEU for 4 years. While at SEU, she has served as Vice President of PPOTA and PHSO, was an RA for Aventura, a Geo leader, a Link mentor, and was most outstanding in her major of pre-PT in her undergrad. Upon graduation, she plans to attend medical school.
Hello, everyone. Hello. My name is Pina, and today I'll be talking about fascia and how it is the body's viscoelastic weight transmission network and its implications for chronic pain and functional medicine. So primarily, what is fascia? So fascia can be looked at as the body's cling wrap in a way. It actually surrounds every single organ in the body, including the bones. And typically it's looked at as a regular tissue or connective tissue, but current research looks at it as a compartmentalization network. But there's actually current research that looks at it as a active weight transmission network instead of a passive container. So first of all, why can we look at it at this view? So say you have a pebble in your shoe, and so the force from this pebble will then go into distant wave transmission networks, distant pain receptors in your body, and it'll travel as facial waves. And so this leads to more important questions like, how do these waves affect distant parts of the body, and what does the body do with these waves? So primarily, before we can get into this, we gotta go to foundations. So primarily, waves are disturbances, and their propagation is driven by the conservation of kinetic energy. And this energy changes from your space and is lost in the environment as heat,
but the energy essentially just transforms, it's not lost. And there are two different kinds of waves that we can look at. There's transverse, which is the displacement is perpendicular to motion, and then there is longitudinal, which is the displacement along the direction of motion. But for today, we're going to be looking at longitudinal, specifically for ultrasounds. So to look at this, we need the physics variables, and primarily frequency is denoted by the letter F, and it's cycles per unit of time, and velocity is the speed of propagation, and wavelength, which is the Greek letter lambda, is the distance between repeats. So it'll be the distance between crest to crest or trough to trough. And period is the time for one wavelength to pass over a given space. So fascia as a closed medium. So this is where it becomes interesting. So we can actually look at fascia as a half closed medium. So on one end, it will be the transducer of ultrasound, which will be the closed end, and then the other space will be the open end, and that's where the nodes can come in. And this is more of an analogy, so it's not like set in stone, but this will allow us to study it in terms of only odd harmonics can go in, so harmonics like 1, 3, or 5, and the frequency for this would be Fn equals n times b over 4L, with n being the amount of harmonics in the closed space, or the half open, half closed space. And then L would be the length of it, and then the wavelength would be lambda n equals 4L over n. So for measuring rigidity, so this would be the shear modulus, which is the measure for rigidity. And the equation for this would be V equals the square root of mu over rho, and mu would be the measure of rigidity over the density. And so this clinical insight gives us the ability to study essentially the different types of tissues. So typically, high rigidity tissues can be indicative of cancer or tumors, versus low rigidity tissues are indicative of inflammation due to their edema. And so in this example case study, let's say we have tissue A and tissue B, and tissue A would have a lower shear modulus, and so this will lead to a lower velocity and lower density, and then tissue B would have a higher shear modulus, which will lead to a higher velocity, and this will, and so as you can see from the graph, we can see that because of the lower shear modulus, we would see that this could be inflammation, versus tissue B could be more of a tumor. And so how can we medically interpret this information? So the transducer, which is the part of the ultrasound that touches you, essentially, it emits waves and detects reflected echoes based on target tissue consistency. And there are 4 main types of ultrasounds, but there are 3 main ones that we'll look at for shear wave simulation that I'll be talking about next. So this is diagnostic, therapeutic, and elastography. And diagnostic just takes images of your body, versus therapeutic, it interacts with your tissues to modify or destroy it, and elastography assesses your tissue rigidity. And so the key thing to note here is that frequency is independent of the tissue, because the ultrasound sets the frequency before you put it on you. And a higher frequency equals better resolution but lower depth. And then the opposite is true as well. Lower frequency equals lower resolution but greater depth. So when we get our results back on our ultrasound, we can see that a hyperechoic tissue would typically be white, and this is typical of normal fascia. And this just means that it reflects back more sound than the surrounding tissue around it, versus anechoic or hypoechoic tissue would reflect back as black, because it reflects back less sound waves compared to the surrounding tissue. And tissues that are gray are typically medium density, so that'd be things like the liver. And its clinical use can be that abnormalities can be found if, for example, tissue like fascia is supposed to be reflected back as white, and it's not, then it can be a sign of inflammation, perhaps. So for descriptive analysis, we can then take these tissues and use Fourier transformations, which just takes it as spatial to frequency domains to then interpret the images as wave motion between pixels. And then for the deformation ratios, we can see that negative ratio would be compression of skin and fascia, versus positive ratios would be an extension of the skin and fascia.
So this is our experimental results of shear wave simulation, which is essentially like using a transducer to promote skin shear modulus. And there has been a 52.4% improvement in the skin after 2 weeks. And for this study, 15 participants were used across different amount of age groups. And it has shown that there's minimal impact on blood perfusion and that it stretches multiple layers of the skin. It's not just one. It stretches the dermis and the hypodermis and the fascia. But its only constraint is that it requires direct contact, so it can't be used on ulcerated or damaged skin. So for the clinical application, we can see that healthy fascia typically has high stiffness and higher wave speeds, and it appears dark if it's fasciitis and thick if it's edema. And we can use our equation mu equals rho v squared for the calculation of that. So in summary, or to wrap up, no pun intended, it is the largest organ transmitting forces as waves, and we can use mechanotherapy to leverage these wave propagations to alter wave signaling and promote skin resilience. And future studies can look into treating chronic non-specific low back pain in patients, as well as using it for biomimetics materials to replace native tissues that may have been damaged. And it can also be used to map body regions to typical elasticity levels to identify pain hotspots and also damaged tissues. These are references. Anything?
So I just want to make sure I understand, but did you say that the tumors have higher wave speeds when you look at them compared to healthy fascia?
Yes.
Why is that?
Because they typically have like a higher density of the tissue in them, like when you look at a tumor, and so they will reflect back more sound waves, and now it would be a higher shear modulus.
And then I guess must have missed one of your slides, but I think it said that SWS promotes healing of the fascia.
Yes.
Yeah, I think that one.
Yeah, so in this study, 15 participants were done. And so another caveat is that more needs to be done, like more people, a bigger range. But they showed a 52.4% increase in their shear modulus, which is just the density or the rigidity of their skin. And yeah, so that was before they did it, that was after they did it. And that's just a general bell curve.
What like frequency did they see to be like the most effective in that?
So the frequency is typically, it doesn't change. So yeah, so the frequency doesn't change because it's set by the transducer of ultrasound, but this would be more of the tested density levels of the patients.
Yeah.
Thank you.
You are finished for the day. If you're in my class next year, you just automatically sign it in.
I did not get the last one.